Right, hello everyone, and welcome to today's BVI seminar. We're doing something a bit different today in that we're going to have a double act presenting. Jolyon Troscianko from the University of Exeter, the Cornish campus, Daniel Osorio from the University of Sussex. Jolyon and Daniel have several things in common. One is that their research has had a big impact on the way people study colour in behavioural and evolutionary biology by importing ideas and methods from vision sciences. The other thing they have in common is strong ties to Bristol, going back well before BVI. Daniel's been a regular visitor to the Ecology of Vision lab in biological
It's about 1993 or something.
Yes. And Jolyon, it's his birthright by virtue of being the son of Tom Troscianko, who set up BVI in the first place. So it's a great pleasure to have them both here, and they're going to help us see through animal eyes.
Thank you. Yeah, and this is new for me and Daniel as well, giving a tag team. So we're not sure how it's going to work. We're going to kind of heckle each other a little bit, and Daniel will take over a little bit later on as well. So some of you might have seen a brief overview of this model before, which has been published, but there's a bit more detail here. And then we get into some new stuff where we take it more into non-human vision, which we think is really interesting. Okay, seeing through animal eyes. So animals need to care about their coloration for all sorts of reasons. So like this nightjar, camouflage is very important. So we need to try and understand how this works. And so the coloration of the animal is critical to its survival and not being eaten. There's all sorts of sexual signaling as well. So the coloration of the male guppy is critically important for finding a mate. And all sorts of other aspects of coloration for signaling and masquerading. So there's a caterpillar pretending to be a snake and so on. So coloration is very important to animals. And anytime you're interested in these kind of evolutionary theories, understanding the behavior or evolution of animals, you need to kind of understand this. And so we need to work out how the vision works, which is surprisingly difficult because there's all sorts of problems with modeling how vision works. So to start, you can make something look lighter or darker depending on its surrounds. So here we've got simultaneous contrast. This bar in the middle is all one gray level. So that gray is identical to that gray. The darker surround here makes it look lighter. So dark surrounds can make something look lighter. But also dark surrounds can make something look darker as well. So here we've got assimilation, the opposite effect. So this gray is identical to this gray, but that one looks darker and it's surrounded by more dark bars. So dark backgrounds can make things look lighter or darker. That's a confusing starting point. But it's not just brightness. And this has been shown in not just humans, but in this triggerfish as well. So this is not due to some human-specific processing. It's not just the brightness of things, it's also the contrast within things. So this target in the middle is identical to this target in the middle. But this one looks lower contrast to you, probably, hopefully, than this one. So the surrounding contrast changes the internal contrast of something. It's not just the brightness, but it's the patterns inside. And likewise with things like stripe orientation and phase and whatnot. So this is identical to this and this, except in orientation and phase. But this one looks paler, like less contrast internally than the other two. So whatever vision model we have should play with these things. You'll see there's lots of stripy and contrasty stuff in nature. Backgrounds are often high or low contrast. These should be really important concepts, but none of the current color appearance models deal with any of this stuff. And that's where our modeling tried to come in. And there's all sorts of other visual illusions along these same lines. So broadly, they fall under the same band of things looking lighter or darker in their surrounds, things looking higher or lower contrast in their surrounds. And the theories behind it, they go from very low level to try and sort of explain from the very early visual processing how this might work. But those models, they're not actually usable by us. They're very difficult to parameterize. And they just don't work. And there's nothing there I could apply to animal vision, for example. But then you've also got very top-down models, which say that, for example, with this illusion, this Allison shadow illusion, your brain is working out where the light is coming from and understanding, all sorts of physical aspects of the scene and using a bunch of Bayesian inference to work out how bright things should be, and then it's not. So maybe that's happening in a hundredth of a second, but it seems unlikely. So can we do it more simply? Now I'm going to pass over to Daniel. You get to explain all of these pathways
Yes.
one by one.
So I think we're just picking up from what Tom was, Jolyon was just saying, but quite a lot of these phenomena that Jolyon's just been describing have been accounted for in terms of various neural processes, various underlying mechanisms, some of which are bottom-up and quite simple, such as lateral inhibition, which can take place in the retina, and some of them are much more top-down, making inferences about the structure of the world and so on. And very often, especially for sort of mid-level ones with orientation and so on involved, they're attributed to quite specific properties of the visual system that we find in the primate cortex. So they're kind of cortex or primate specific. And that means that it's rather hard to know how they might be relevant to organisms with different kinds of brains. So let's have a look at the next slide.
Bring in the animal.
Oh yes,
brain
and
bits.
so we have different animal brains here. So shall I just take over advancing the slides
No,
here?
you can use the down arrow on that.
That's fine. Yeah, so there we are. There's all sorts of different vertebrates there, with more or less lacking any kind of neocortex. So another way to think about neural processing that was emphasized historically very strongly by Horace Barlow was a principle of efficient coding. So he was interested in understanding especially the properties just of the retinal ganglion cells that center surround neurons that send signals from the eye to the brain, and he put forward 3 hypotheses about the functions of these neurons, how they could be interpreted as matched filters looking for particular objects of interest, for example, was one of them. But far the most influential one, and the one that Horace thought was correct, was this idea of coding efficiency. So essentially the principle here is that you want to pack as much information about the visual world into a relatively low bandwidth optic nerve, or costly nervous system. And to do this, you essentially have to strip out redundancy. So you try and have a code in which the signal at any point of space or time cannot be predicted from the signal at any other point in space or time. You move redundancy, and you also use your bandwidth efficiently. So these are the principles of coding efficiency. And this idea, about 20 years after it was originally proposed by Barlow, by Simon Laughlin, who was effectively my PhD student, PhD supervisor, showed that these principles of coding efficiency could be applied to understand early neural processing in this fly. And what Simon did was 2 things. He went out and measured the statistical properties of natural scenes, so he knew what the input statistics were, which is essential to any model of this kind. And he measured carefully, as we can see on the right here, the actual physiological responses of the second-order neurons, the neurons that are postsynaptic to the photoreceptors in the fly's eye. And essentially he showed that initially, in the first piece of work he did on this at least, that the contrast response function of these cells was matched to the range of contrast that they would experience in natural images. And what that meant was that each response level, each gray level in the output of these neurons was present equally often. So it was making full, efficient use of its dynamic range. And they then went on to apply this idea to spatial coding as well. And when he originally published this work in the 1980s, it was published in an obscure German journal. Almost certainly I never asked him because it was rejected by Nature. It's quite interesting. If you, what not completely ignored, but in the 1990s, there was an enormous explosion of work on coding efficiency in relation to properties of the cerebral cortex, V1, and so on. But it's only in the last 5 or 10 years that people in the field have actually come back to recognize the significance of this very simple, easily understandable work on showing that a nervous system actually produces an optimal code for natural image statistics. So that's the principle that underlies Jolyon's model. And another point I wanted to make, just in case you're not aware of this already, is that real neurons, which are producing action potentials, have a very limited information capacity. They can only produce spikes up to a certain rate. Producing these spikes is metabolically very costly. It uses a lot of energy, so they want to spike as little as possible. And if we're thinking about any real nervous system, it can spike up to about, say, 100 spikes per second. And the integration time that we have in our vision, say, is probably about 100 milliseconds or so. I'm sure there's people in the audience who know that number better than I do, but it's something like that. So we don't have that many bits per integration time that we can encode with a single neuron, and that is relevant to the number of gray levels that we use in the model, as we'll see in a moment. And so this just reiterates the points that I've made before and underlies the model. And I think with that, I will return to Jolyon.
Thank you, Daniel. Yeah, so when we came to think about this model, here's some of the basics of the model. So first, we assume the image is encoded by these neural channels of equal bandwidth. So the same neurons of the same bandwidth can convey the information from the eye through the brain, and so on. And this is an odd one that I'll go into more detail later. The dynamic range of each channel is a fixed multiple of the lower threshold. So if you've got a neuron that can only fire between 1 and 10, at 10 highest and 1 lowest, well, that lowest has got to be this lower threshold, and the upper one is then a multiple of that. It kind of stands to reason. And then you need to sum all of these channels back together again, and we do that in a way that makes sense with natural image statistics, going back to the Fly natural contrasts model. Yeah, so this has been published, but we're also going to go into some new stuff that we've done as well. So the first thing the model does is break the image down into different orientations and spatial frequencies. And so, yeah, it can use non-oriented filters as well. Actually, it doesn't care in our model whether you use center surround or Gabor oriented filters, weirdly. They're both pretty good. So you take the input image, you output all these. So this is the highest spatial frequency and the lowest spatial frequency. Split it up like that using, difference of Gaussian or Gabor and so on.
So these are octave bandwidth?
Yeah, 1 octave bandwidths, which seems
very standard.
It's standard. It's a sensible thing to do. And so the output of the model goes like this. So if the x-axis here is the contrast that you see, so with your center surround contrast, for example, what is the output of the model? Well, first you cut out everything that's sub-threshold. So the contrasts that are too small to see go to 0. And then if you assume that your neuron has a bandwidth of 4, so it can fire, between 1 and 4 hertz, then you take that that threshold and you multiply that by your bandwidth, you get up to here, and everything above this is saturated. So contrasts above this level all become saturated. So you get this response function. Only these green ones are actually coded for dynamically. This is your dynamic range. This is saturated, that's lost. And then you repeat this for all of the different spatial frequencies, so on those octave levels. So here we've got some different contrast sensitivity functions. people know what contrast sensitivity functions are. On the x-axis, we've got the spatial scale. So this is very fine things, and this is very coarse things, low spatial frequencies. And this is the sensitivity. So humans here, we've got a sensitivity of about 200 to 1 at 2 cycles per degree. And that's massively better than pretty much anything else. And here I've plotted the graph in the same function as this, same way up, upside down if you like, showing that at our peak, we've got this with a bandwidth of 4, neurons that code for 4 bits, very tiny dynamic range where we're good at seeing contrasts, but things saturate very quickly. Whereas at the lower and higher spatial frequencies, we get this enormous dynamic range, but very low sensitivity. So there's a trade-off between those two. And if you take these, that's basically all a model does. But one thing that we don't know yet is what that dynamic range, what the bandwidth is of the neurons. And that's not straightforward to work out what that should be. And one way that I tried to work that out is from this crispening effect. So this is a really interesting visual illusion. This row of gray squares is identical to this one, is identical to this one. But what you should see is that on a light background, the contrast, the difference in steps between these ones should be quite high, and these ones quite low. But here, that's the opposite way around. Here, these steps are very large steps in contrast, and these are small steps. So the contrast is dependent on the background brightness. And luckily, Whittle, back in '92, did some very painful, laborious experiments to actually model this horribly nonlinear function with different background colors, getting people to work out a uniform contrast step space. And then all I did is fit my model against Whittle's data. So you get the lines here showing Whittle's data and fitting either my model with a difference of Gaussian, non-oriented, or with Gabor filters, the oriented. And it just spurts out a number of what it thinks the bandwidth is, which would be 15 for a difference of Gaussian, or about 4 for oriented for Gabor filters. And that's really nice. If I take those same numbers and put them into these data of a macaque magnocellular neuron ganglion response firing, it actually fits better than the fitting curve that the authors used themselves back then. So this implies that for example, a bandwidth of 15 actually fits from bottom up. It fits the neural responses from bottom up data and behavioral data top down, the crispening behavioral data. They both all suggest that you've got this bandwidth of about 15 to 1 for the neurons, which is quite neat. It's nice when you've got these kind of things linking up. So we split up the scene.
Sorry, just to clarify, what's that saying is that the dynamic range of the neuron is essentially optimized for coding information, given that it's limited by some sort of internal noise you can't get around that sets the basic contrast threshold. This is of course dependent on the spatial frequency, on the area of retina you're integrating over or whatever.
There's other neat things, like it's no coincidence that non-oriented is about 15 to 1 and oriented is about 4 to 1. It does it at 4 orientations, and so you multiply that 4 by 4 and you get just about 16. The same information you can put through lots of oriented filters with lower bandwidth or higher bandwidth center surround. So we split the scene up into all these different spatial frequencies. How do we recombine them? This has been a big problem with similar models like this. We just assume that this weighting, the way that you weight the model, is based on natural image statistics, going back to the Simon Laughlin thing. All of these neurons coding for these different spatial frequencies should all be firing, roughly equally. And so we whiten the image so that in a natural scene they're all kind of behaving similarly. Another way of viewing this is you take this image, split it into its spatial frequencies, apply the model, and then the weighting that comes from each is kind of equal for a natural scene. And you get this weird output that looks like an impressionist painting, which is quite cool in a way. In a way, it's nice that this is an internal representation. This is not meant to look like a nice picture to you. This is what kind of might be going on inside your brain. But the fact that artists come up with similar looking things is quite neat. And so that's the rationale for pooling the output. But a really interesting thing about this model is how much information is thrown away. In a visual scene, there's going to be contrasts all over this contrast space, spatial frequency contrast. But we're throwing away everything in blue and everything in gray. How can we throw away so much information without causing some weird artifacts, compression artifacts? Like, JPEG images have little, square artifacts that you get. There must be an equivalent for this massive compression of data. So one answer to that is actually there's enough overlap between these bands that actually it's not as bad as you might think. So there's a bit of, because there is some overlap, if we take this image of lots of spatial frequencies, put it through the model, and this is showing the output from each band, there's enough overlap between them. All the blue is the saturated, all the gray is the invisible. There's enough of this green going on at different spatial frequencies that actually you can sort of see everything, and there is a bit of redundancy that can cope with it. And this helps to explain how we can see we think we can see such high dynamic ranges. So when you go out and buy a modern TV that's high dynamic range, it can have dynamic range of over 10,000 to 1, 100,000 to 1. How can we see that with neurons that can only code 10 to 1 contrasts? And the data for sine waves show we can only see maximum 200 to 1. Why are we buying these TVs with 10,000 to 1 contrast? And our model can kind of explain how. We think we can see those, but we can't. These compression artifacts are actually coming in. So to some degree we can, but in most degrees we can't see that those contrasts, which is quite interesting. But also those compression artifacts, they come in here with all of these visual illusions, because we tested over 50, basically all the standard luminance visual phenomena. And broadly, it explains the direction of all of them. There's a couple that it doesn't do very well, but basically everything you see here, it does a really good job of explaining exactly how this works, which is quite neat. So we can go through some basic examples. This is Adelson's shadow illusion, where this gray square is identical luminance to that one, but that one looks lighter. And yeah, sure enough, the model can do that. This is actually quite an easy one to do with low-level modeling. Where this gets more difficult, this is kind of simultaneous contrast. Where it gets more difficult is working out how the simultaneous contrast weighs against the white illusion. And it can sort of reconcile those quite nicely. And so going back to this crispening example, this can show us why. Here's the output of all the same image at different spatial bands. So we can actually work out why exactly we think something is as bright as it is, because we can interrogate this and see where is the saturation happening, at what spatial band are we losing information because it's too low contrast to see or too high contrast to see, and therefore being got rid of. So if we zoom in here, where is all these green ones? So you're really good at telling contrasts here, the white against the white background, the darks against the dark background. You'll see here, there's lots of green going on there. Your brain is working very efficiently at coding these ones, and these middle ones. You're very good at seeing these contrasts and these contrasts. But outside of that, all these blue values, they're meaning that you're reaching the saturation. The contrast is too high for it to be coded for, so it's just saturating out. And so it's appearing to be lower contrast than it really is. Yeah, so this clipping can explain it. And this, again, helps to explain why we think we can see super high contrast in nature, but actually our eyes just can't quite code for that. Other examples like this, how do you pronounce it? Chevreul staircase. So for example, look at this low spatial frequency, there's gray going on this bottom one. So people who don't know this step here is identical to this one. It's just that you take that and twist it around 90 degrees, so 180 degrees, it looks like that. So instead of looking like a staircase, it now looks like a series of gradients. And again, the model can explain why. This is sub-threshold at one spatial frequency and meeting threshold at another. So it meets the threshold in these spatial frequencies, but it's sub-threshold here. And that's why this is happening. And so on. Another fun thing we can use for a model is to shove color through it and see what happens. And something that has really annoyed me as someone who studies animal vision is the von Kreis assumption. We always measure a white standard or a gray standard and assume that your visual system can work out the color of everything in the surroundings from that one white or grey measurement. And we know that's not how visual systems work. The color of anything depends on its surrounds to a large degree, as well as the global illuminant. So it's local versus global illumination. And there's been studies kind of quantifying this and showing it's just a difficult mess, basically. So anything we can do to reconcile this would be very exciting. And so this is quite a nice illusion, Lotto's cube, but with yellow versus blue filter. What to you probably look like grey squares on the top, sorry, blue squares on the top here. They should look blue to you, and the yellow squares on this side, they should look yellow to you, but they're actually an identical grey to one another. So this is local color constancy failure. And the model can kind of deal with this really, really nicely. It thinks that the yellow looks yellow and the blue looks blue on this side, and it thinks the yellow looks yellow and the blue looks blue on this side. So it can kind of reconcile this local color constancy effect, which is nice. And it can do the simultaneous contrast illusion as well. So this square is identical to that one in the input, but this one looks a lot brighter because it's in the shadow, simultaneous contrast.
And of course, the way this illusion is presented, you're encouraged to think that this is due to various kinds of high-level knowledge about the illuminant and the orientation of the surfaces and so on. So this is an entirely bottom-up.
With a super low-level model, you can explain this, barely past the retina level neural processing. You can start to explain this. But wider chromatic, like whole scene chromatic adaptation, not just color constancy. And a really interesting thing, if you take this image of a woodland, multiply the red channel by 5, multiply the blue channel by 5, shove it through the model, you get these outputs where the green always looks green and the bluebells always look blue, whatever you put through it. And this is really interesting. When you walk through a forest, you hold out a gray sheet in front of you, it doesn't look bright red like it should. If it's global illumination, which is green all around you, it's really difficult to work out why a white thing in front of you doesn't look red. But this can deal with that in a purely feed-forward way. There's no feedback loop in the model that measures anything or averages anything. So that's a really nice aspect of the model in a way, that you can kind of reconcile chromatic adaptation, global and local color constancy, maybe to some degree. Needs more work, but the idea is there that with a purely feed-forward, like single neural layer connected to another neural layer with hardwired neurons, you can get this effect. There's no scene averaging of any kind. You've averaged the model originally to natural scene statistics. After that, there's no model change. Okay, so that's all. That's kind of the basics of the model. And now I'm going to sort of go into animal vision a bit more, which Daniel and I are working on a lot more at the moment. Because ultimately, that's what I want to do. I kind of don't care about human vision. I kind of do, I guess it's important. But I'm a biologist with evolutionary theories, and I want to be able to model what the world looks like to a non-human. Everything's worked on color so far. There's so much work on avian color vision and bee color vision. Everything's focused on color. But actually, most information in the world is luminance. When you build a television for the first time, you don't give it the blue-yellow channel or the red-green channel. You give it luminance. Black and white TVs were invented first. And they're really good. You see almost everything you need in a black and white photo. Color only adds a little bit more. But luminance is where we're crap at dealing with stuff. And if you look at the contrast sensitivities of non-humans, it goes crazy. So us humans, we've got contrast sensitivity of peaking at sort of 200 to 1. Whereas most birds, it's 10 to 1. So our contrast sensitivity, 20 times higher than the birds that we're looking at. And our visual models that we use do not reflect that. They kind of do a bit in terms of the color. But when it comes to luminance, we're not kind of dealing with it at all. So if we take our model and put this shark, this is a Port Jackson shark, through it, the human model, we get this nice output, and you can see how it looks to a human. A really interesting thing, so humans have this high contrast sensitivity, but a lot of fish have very high as well, which makes sense. If you live in a murky environment, there's an arms race for seeing low contrasts. And so if you have this Port Jackson shark, very low acuity, so can't see much detail, but actually quite good contrast sensitivity, and a little blenny, slightly better than the shark, so higher acuity, higher sensitivity. But then if you get a chicken and you drown it, apart from it being quite unhappy, it really can't see that much detail. Even though the chicken has much higher acuity than the shark or the blenny, it just can't make out those contrasts. It's got such low contrast sensitivity. And our model kind of suggests this interesting trade-off between if the same, so your neural bandwidth, neurons can't fire any faster than they can fire. There's a physiological limit to how fast a neuron can fire. So if you want fast vision, because you're flying around and catching fast things, you need to pool for a shorter time. So you need a smaller bandwidth. So birds with fast vision can't have as high neural bandwidth. So maybe that's why they have this much lower contrast sensitivity. Don't know, but our model would imply there should be a trade-off, which is quite neat to explore. Yeah, and so another interesting thing that happens is you get recovery of contrast in the fog. If you look into this kind of foggy distance, not only can you see more details as it gets foggier and foggier because you get contrast enhancement with more and more fogginess, you get chromatic constancy as well. So even though the water is making everything murky and blue, we're getting chromatic color constancy. The color of these algae is the same at different distances up until it all collapses. So we're getting natural kind of chromatic adaptation, a color constancy through a material that's colored. And yeah, the model doesn't work very well for non-human chromatic vision. We don't know the parameters very well, but you get the same idea. Even a blenny would have quite good being able to see that algae is red versus the ones over there green through the murk. That's a really murky picture, and we're recovering this color information.
Very good contrast constancy there.
Yeah, look at that contrast constancy. It's not fading out gently as it does in, the input fades away nice and gently. That's actually just, you suddenly reach a point where you can't see anymore. Really interesting. So we're getting a lot for free in a way. And this links to that phenomenon I showed earlier of this textural contrast induction. Makes sense. It's a nice kind of trade-off. So one thing our model needs is a bank of natural scene statistics against which to kind of whiten your image to work out how to pool your bands. And this is just a little illustration to show it doesn't matter that much. So I took this Port Jackson shark model and put it through a few benthic images, very low contrast, pictures of murky bottom of the sea kind of horrible stuff versus lovely woodlands, and you don't get huge differences. So even though these are quite dramatically different visual scenes with quite different functions in terms of their image statistics, the model kind of spurts out similar stuff with either of them to be explored, but I suspect there's not going to be a huge amount there. It's going to work quite neatly in different systems. So the state of the art in most animal vision modeling at the moment is just to think about the acuity limit. So when people are looking at a caterpillar and want to know the distance at which these bright, yellow and black, sorry, this isn't a color image, these yellow and black bars become invisible to the bird or whatever, there'll be a distance at which this becomes camouflage. Innes has done, amazing stuff on distance-dependent camouflage. At the moment, we just use acuity. We work out the spatial frequency at which you can't see any more detail, and we kind of just chop off the information above that. So that's kind of what people do at the moment. But if we use our model, we get really different effects, as you might imagine. So if we actually model a contrast sensitivity function, you get this kind of thing. So this is chicken vision at the same distances. Sorry, I didn't say this is viewing distances at the top. So when you're up close, 10 centimeters away, the chicken can see all these bars very easily. But from nearly a meter away, you can barely see anything at all. So if I flip between those two, you can see there's quite a lot of differences going on. But another really neat thing about our model, which makes me very happy because I'm now working quite a lot on the impact of artificial light at night, is we can see how it looks at low light levels. So people have measured the contrast sensitivity functions of chickens at low light. And so now we can just fit that lower contrast sensitivity function to our model and see how it looks. And this is really good because the existing low light vision models that we have for animal, I mean, they don't really exist. The chromatic ones exist, but they're so difficult to parameterize. It's insane. But here, you need very little information, and you'll be able to work out what the world looks like to a chicken in different light levels, which is quite neat. And it makes a dramatic difference, as you might expect. So here's all three together. So there's the kind of acuity only, and then standard contrast sensitivity functions with our model. and then low light version of the same model. And so that caterpillar really starts to become invisible to the chicken under this kind of low light.
So somebody's measured the contrast sensitivity of a...
Someone has, yeah. People in Lund do all sorts of crazy stuff. I think it is Lund people, it's bound to be. They've done it with owls though, and fish, actually, and it's such an easy thing to measure with an animal is its contrast sensitivity function. There's all sorts of interesting stuff you can do in terms of distance dependent effects. So if we have this poison dart frog and see what it looks like to a human can see it at pretty much all distances. The patterns look quite nice until it just fades away to nothing. Whereas with a kestrel, with very different contrast sensitivity curve, we're getting some really interesting phenomena. So here, look, the head of the frog is suddenly really bright. This dark surround in the background is having a simultaneous contrast effect. Whereas now the head is looking really dark. The dark surround is making the head look darker. So if we trust our model, which to be fair is a big if, human vision is really, really different to non-human vision in that sense. Because we have such high contrast sensitivities, we've got such a high bandwidth of information throughput to a bird that needs fast information, and it must have lower bandwidth. And we know that it's got much lower contrast sensitivity. There could be all sorts of visual phenomena that are much more common to it than to us. So that, it's a very different world to them. And we can model these effects, of course. So we can look at how to human vision. So blue human vision here with distance. If we view something high contrast signal from a distance, we get quite nice stable drop off with distance. Whereas a kestrel looking at the same thing, there's a certain distance at which this moth looks absolutely insanely brightly colored internally. But just one specific distance, and that will be dependent on properties of its surrounds and all sorts of weird stuff. And so, it's quite difficult to model. But human vision is probably more stable than non-human in many situations like that. This is kind of showing the same thing, how if you have a gray background, so if you have the frog on a gray background versus a natural background, it looks very different, a bit different to humans, but the non-humans is even more dramatic. So if you take a frog and put it on a very plain background, it would look more different to a kestrel than it would to a human at certain distances. So in terms of understanding how animal signals work, this is potentially quite important because it shows that these distant dependent effects are working quite qualitatively differently in humans and non-humans. Which is quite fun. Just a final thing is a really nice output of our model is all of these band pass layers. We've got the same scene that's put through all the spatial filters, different orientations. And this leads itself really nicely to put into a salience model. So this salience model, it takes the center versus surround of those bandpass images that are output from our model. And it sees whether what's going on in the middle is the same as what's going on in the surround. So it's a sort of second order pattern effect. If you get this difference in orientation in center versus surround, the salience model goes nuts. If you get a difference in spatial frequency in center versus surround, it also goes nuts. So it's very sensitive, but this is using our output model of only the contrasts that the animal can see. So we can take this and take, it's just really nice and easy to put images through this and see what things look like. But is this useful? Well, who knows? But so it's nice to peg this against some behavioral data. So that's what I've briefly done here. I'm sitting on, of course, big piles of data of camouflage where I've got humans to poke touch screens trying to find moths like this. So each of these has a little artificial moth here. And you can put it through the saliency model and then see how salient that moth looks. And you can see it's doing a really good job. Some of these are really well camouflaged. And the saliency model is always picking out the moth, or at least the bit of the moth that seems most salient. So I was really hopeful that it would be very good at predicting human performance. So I had this old data set. And this is a horrible graph showing the performance of different camouflage metrics to predict how well people would find camouflage things. This was a paper from a few years ago where this edge disruption metric was really good at predicting stuff. And if I take that saliency model, this is its output. And I was a bit sad that it wasn't higher. I was thinking, it looks so good. It should have been really good. But then I realized that all of these things above it, these all have a lot more information. In fact, pretty much everything of here they're given an advantage over the saliency because you're measuring the moth against the background. So it's got knowledge of where the moth is and what the background is before you run the model. Whereas actually this one doesn't. It doesn't know anything about that. So it's getting very good performance without knowing as much about the underlying. It's not told where the edge is. This thing, I tell exactly where the edges of the moth is all the time, whereas this in the analysis, it doesn't know. So HMax, for example, is doing almost exactly the same thing, but that hasn't passed through our visual model. The HMax is looking at Gabor filters at different orientations and comparing how similar the center and surround is. And it performs fairly poorly. Well, it was okay, highly significant. But if you take our model and look at its salience, it's 3-4 times better at predicting human performance. So hopefully that's making it quite a useful thing and means that this saliency thing could be useful. It's a really easy metric to output from a model that could be useful for biologists or other people interested in non-human things to use. So that's it. Here's a summary. So accounting for all sorts of interesting lightness and color phenomena, at least the direction of the effects, if not fully quantitatively, but qualitatively it predicts the right directions for loads of effects. This super low level efficient coding could be responsible for a lot more than we previously thought of. I really like that. No, you don't need, you can have color constancy, chromatic adaptation with no need for adaptation or normalization or divide by the average kind of things. And hopefully now we can make it easy to apply it to non-humans. So that's our next thing that we're going to publish soon, maybe, hopefully, is making all of this animal vision stuff really easy to use. You'll only need to put in the contrast sensitivity function and basically everything else it works out for you, which is nice. So that's it. Thank you.
So, questions for our speakers? Cedric.
You said all you need to plug in is the contrast sensitivity function, whereas with conventional current state animal vision modeling, you have web fractions, MSP data, spectral sensitivity data, a lot of decades of work that goes into a specific animal vision system at times, which leads to many people sort of using proxies from all sorts of Frankenstein visual systems. Are you implying that this general issue tends to be alleviated with this way of thinking about it? You need to parameterize things?
Well, a really nice thing about the contrast sensitivity function is it takes into account all of that neural noise, inherently. It's accounting for neural noise and photon noise, all of these sources of noise. It's kind of, to some degree, accounting for already. And that was one of the rationales for, I wanted a model that I could model how the world looked to an animal with some behavioral data that was quite easy to extract from the animal. So I'm sure it will be wrong, but it's at the moment there's no luminance model that works even close that that can deal with these phenomena. Your work with the triggerfish and their luminance, basically it didn't work very well. But if you were to take that, if you were to maximize those visual illusions, put it on a slightly darker background, everything would go out the window. So we need a model that can deal with that. So it's a starting point, hopefully.
I guess the point I'm getting at is, in addition to spectral sensitivity.
The luminance channel that you put in should vaguely.
Thing that we which is the contrast sensitivity.
Jolyon's model does also take account of the contrast sensitivity functions of the different chromatic mechanisms, which are known for humans, but
not for non-humans.
known for anything else.
So, for example, those contrast sensitivity functions, very well known for luminance, but you can make those same functions for the chromatic channels as well. So that's what we would ideally want. If you want to use this model for color vision, that's why that Blenny picture was so crap, because no one has measured the chromatic contrast sensitivity of Blenny vision or any non-human, to my knowledge. But that's what we need. They should be easy data to get behaviorally, at least easier than MSP and all of these other techniques. But no one's done it. Who knows?
How would you get that?
You sort of show a screen to the animal and get there are different ways of doing contrast sensitivity. You can put it in an optokinetic optomotor drum where you can, so you'd have red versus gray bars instead of white versus black in a sine wave. And you change the chromatic difference between the peaks until it needs to be isoluminant though. So it's a bit tricky.
What's the reaction?
But in a drum like that, if it moves its head or its whole body with the thing, then it's, or you train the animal to always go for horizontal bars instead of vertical, and then it has a two-choice thing. There's different ways of doing it. It's that kind of thing. You can do it with fairly simple, either operant conditioning, but even better in optomotor, because then you don't need any conditioning at all. So contrast easy, but chromatic not maybe.
Thanks. Yeah, awesome. I guess with optomotor drums as a behavioural assay, you've got motion vision that is taken into account there. So there's a step of processing. We know that for a lot of animals, they'll only use a single channel of spectral sensitivity for doing that behaviour.
And not always, and sometimes a lower spatial frequency than is their visual limit. Absolutely. So I guess that's it's not ideal.
Factors with that as a behavioural assay.
Yeah.
And that's where maybe conditioning trials and so on.
Yeah, definitely. But yeah, no one's really done them at all, like chromatic side of things.
Which suggests it isn't so easy.
Maybe. Well, proving that it's isoluminant would be difficult.
Well, we're running quite a lot of that with crabs at the moment, and if you have green and blue stripes, you can balance the brightness of those stripes to get the zero response, so it's completely isolated.
Really? Okay. Yeah. That's perfect.
But you can show a crab a looming stimulus at that same isoluminant point, and you do get a response.
Amazing.
So it's a different motion.
So if you could plot the spatial frequency of that function, then you could shove that into our model really easily. And interestingly, the chromatic human ones, it's basically a step function. Everything below a certain spatial frequency, you're sort of very good at seeing the contrast, and then it dips off. Whereas you don't have this inverted U shape, which you have for luminance, to my knowledge.
That's right.
So with the poison dart frog example that you showed towards the end, if I understand it right, that the idea is if I'm a kestrel and I'm flying towards a frog, its head should be
flashing
like a lighthouse. Like a beacon.
If you believe the model. No, but actually, like the only contentious thing our model is doing maybe there is cutting off that threshold, the upper threshold, where you're cutting away information that you can't see. That's not having that is doing something in our model, but it's only doing a bit, and you can chop that out and see what that's doing. The rest of the model, just applying contrast sensitivity functions, that's not very contentious. So, and that still does that kind of flashing effect to some degree.
There's a lot more information going on that we are not sensitive to. It's not just say that you blend together from the distance and your warning color when you're close. A whole story could be going on.
As you approach, when you've got such cheap pre-processed vision. So if you think about bird vision, they're squeezing, their oil droplets, their color vision, everything is pre-processing their color vision to the extreme at the retina kind of level, and compressing it so much that it's easier for the brain to deal with. Maybe that's one idea. Whereas our human brain is just so vast, and our visual, those pathways that Daniel showed, even though we've got such overlap on our red-green vision, we still have better red-green vision than birds without any of these oil droplets. So how's that happening? Neural stuff, so if birds can strip away that neural thing and get cheap, quick vision, there's going to have more artifacts, more compression artifacts. And we would expect evolution to tease them out and play on them. So that frog might evolve to do exactly that, to look like a flashing thing as the bird approaches it. Who knows? But it kind of gives you the idea that maybe the way we see the world is really actually quite different to a kestrel zooming in on a frog.
Thank you.
It has been extremely interesting. I'm wondering, all these models that you have created and the contrast sensitivity functions, are they based solely on still images?
The contrast sensitivity functions I've taken, they're all just sine waves. Sine waves of different amplitude, basically, for humans and non-humans.
Is that what you mean?
What I'm wondering is whether, I thought that the contrast sensitivity function, you basically show contrasting colors within the pattern of the sign. And then you basically try to record the reaction of either an animal or a human, right? So what if it is moving? are we able to perceive the same levels? Or are we, are we basically even further compressing?
Motion vision and how that ties in.
Yeah, exactly.
Yes,
With
good
contrast,
question.
yeah.
I mean, I'm working on motion vision stuff a fair bit at the moment, but it's yeah, who knows? But
in general, the same principle
exactly
will apply to.
We should be using
moving
similar.
stimuli. You can think about the receptive field of any neuron as a spatio-temporal structure, of course.
So my motion vision models now, I'm taking the pre-processed output from this model, and putting that through the motion detectors then, because that seems like a sensible starting point. Your retina's got center surround stuff going on, and it must be limited. There must be, upper and lower thresholds at which things can and can't code.
Well, right?
Oh, what? Sorry.
There's also a memory effect. I think.
Yes. Yeah. Well, for motion detection. Yeah. Absolutely. Yeah. And you can discuss how that might work in non-humans, but you're right. Like this should all play into the underpinnings of all visual stimuli, like not just color but also motion. Yeah. Maybe, but motion's just an odd one sometimes, isn't it? Get Nick to.
So what are the selection factors that have pushed humans to both very high spatial acuity and high contrast sensitivity?
Yeah, I don't know.
Better than both Blenny and Kestrel combined.
And if you think about it this way, so there's like a handful of species with higher acuity than us, but really only a handful of birds. But they've got awful contrast sensitivity. So our vision just absolutely canes everything else in nature, if that's true. So one problem is maybe the contrast sensitivity functions that I showed for humans, that's because it's easier to measure in humans because you can talk to them and you can give a cup of tea and put them in front of a computer screen. And maybe we're not just maxing out what the non-humans can do. I don't really believe that. It's been done so many times and we're always so much better than everything else. So if you assume it's not an artifact, if it is true that we've got this high contrast sensitivity, why? I mean, one idea I had that I was discussing earlier was, well, we've got such vast brains for something.
Well, it's to deal with all this extra information.
Well, maybe, but let's assume it's not for visual information.
But it is. Our brains mainly vision.
Well, in that case, that's, I guess, is your answer. We're hunter-gatherers, and we see better than everything else out there.
Can I challenge that, though? I think that quite often it's because we haven't asked the right question of the animals when we measure them.
Yes.
The work that we did on cuttlefish was a prime example where they saw 20 degrees difference in polarization until we asked a better question.
Yeah.
And then they were seeing less than 1 degree.
Yeah.
So I think that a lot of the experiments we've done, we've made assumptions about the way we test them, optomotor results, other types of things.
Yeah.
And it's the wrong question.
Yeah.
And I think if you were to try multiple different things, think about their umwelt and what are they living to see and do, and apply that to the way you test them, you might suddenly find that actually the results are far better. I mean, it doesn't make any sense to animals that have evolved for as long as we have. Would have for the cuttlefish and octopus, for example. To think that 500 million years of evolution only gave them 20 degrees difference in polarization didn't make any sense. Less than 1 degree suddenly makes sense. Our color vision can see differences of less than a nanometer, which is unfathomable when you make it as a measurement in color, right?
Yeah.
Other animals have 4 or 5 different receptors. They have different receptors in different parts of the retinas, and those are all doing very different things. And are probably tuned very well to what they do.
Yeah.
We're just not measuring it properly.
That's definitely going to be partly the answer. But also, like, the acuity, we know that we're not that far off with contrast sensitivity functions because we know that the acuity, ganglion counts in the retina, they can't be that far off. We know the retina can't be doing much more than, I don't know how easy it is to go back to a relevant example. But from, here we go. So from ganglion counts, we know that the acuity isn't much higher than that because the retina's just not supporting it. So from this acuity point, actually, that U shape following it, that kind of makes sense.
No, the acuity values are pretty easily tied to physiological data. It's the contrast sensitivities a little bit more.
So the space here is probably fairly robust. The height here is what is really in question. Yeah. So can we do behavioral experiments that show that actually the fish can see? And you should expect there to be an arms race in fish, sharks, for seeing in murky water. seeing these very fine contrasts of things looming against you.
It's how you tell distance underwater. That's something changes contrast. That's tiny the distance that's changing.
Yeah,
You
yeah.
can't tell outside, 'cause you
Well,
don't know what it's
unless your
doing.
retina is correcting that for you without you noticing to some degree. Yeah, yeah. But yeah.
But of course, to go back to Ulyss's question, we have a very small fovea, so this high spatial acuity is restricted to just an angle of about a degree across or something like that. So if you compare us to other mammals, for example, in terms of overall spatial information, the difference is not as great as it might seem.
Yeah, I was going to follow up on that. I mean, why don't you have any primates on that graph except humans?
They'd be very close to us.
They are very close. Yeah, they actually
Very,
just sit
very
on
close
it.
indeed.
Yeah.
So it's not really hunter-gatherers that does it.
No, it's primates.
Well, yeah, they're generalist
The art is, and the
feeding. Right?
performance is also very strikingly independent of our size as well. So we're about the same as a marmoset, just with the basic optical scaling factored in. The packing of the photoreceptors, the size of the photoreceptors is the same in a marmoset as it is in a human, which is at a fundamental physical limit. You can't make photoreceptors any smaller than they are in a graph of the air.
So he shifts that way a bit, but stays high. If you're a tiny primate.
One thing I really wanted to highlight in this model, especially since we're in the psychology department in Bristol, is that just how weird it is that people such as Richard Gregory and many since have highlighted the idea that what our brains are doing is recovering some sort of veridical experience of the world based on limited information. So we've got sort of prior knowledge and top down and all that sort of thing. And what Jolyon is saying with this model is actually what we're seeing and what we're accounting both for the appearance of these natural scenes, perhaps, and certainly all these illusions, can just be explained in terms of packing the maximum information from our retinas, from our photoreceptor array, and getting it into the brain. There's nothing else there.
Yeah, that biggest bottleneck in the system probably should have a big impact. Yeah.
Yasmina had a question.
I wanted to actually ask about this. What is the proof of category of illusion that you model really well? Is
no. Yeah, it's quite nice. You can take the white illusion, for example. You can set up a simulation like this where you change it very slowly. You change the background. So you start with it very similar, and it gets darker and darker, and the simultaneous contrast kicks in first, as the contrast is quite close, and then you can plot that inflection point when it becomes white illusion. So you can do all of that. So it goes sort of seamlessly between the two, and you can predict exactly the viewing distance and the contrasts at which you will switch from simultaneous contrast to the white illusion. It can do that. Whether that number is accurate, no, it won't be. It's not perfectly, calibrated and parameterized, but it's pretty good. It's pretty close. So in short answer, quantitatively, it's impossible to test. People haven't got the right data in most situations for us to quantitatively test it. That's why that whitening example, the crispening example from Whiten, the crispening example, it was quantitative data. So that meant I could fit the model to it. That was just some behavioral data, top-down data to peg it all on. And it fitted it. As you can see, it fitted that shape of the model really nicely. And that's why the crispening effect is so nice for that, because it does take into account white illusion and simultaneous contrast and other contrast effects, all in one kind of weird illusion. So if you can fit that, my hope was at least you're getting close to fitting some other stuff.
Last one from Martin.
Thanks. Yeah, your comments about the spatial acuity side of contrast sensitivity curves being quite well defined based on anatomy and it matches up with anatomy.
Well, at least gives you a upper number, if not a very reliable
It
one.
also got me thinking kind of along the lines of Simon Laughlin's thoughts of how photoreceptors effectively turn signals into noise based on the number of microvilli in a fly eye or something. Could you equally generate anatomical predictions for the rest of that curve based on the photoreceptor size, how much photoreceptor space is invested, how much opsin is in each one, and
To get out the contrast sensitivity functions.
Try and backtrack.
Oh God, I've no idea.
Sounds like the kind of thing that Simon Laughlin's probably thought about quite a lot.
Because it wouldn't be worth having photons that could code for this bandwidth unless you had the neural architecture afterwards to deal with it. That's really nice. Why I really like it, that the number for that crispening fitting, that roughly 15:1 for the non-oriented and 4:1 for the oriented. That's what happens in your brain. You know, it starts off with non-oriented information from your retina, and then in V1 and stuff, once you get into the bits of your brain that do orientation stuff, you can then get away with smaller bandwidths for the same input. So the whole system should be geared for that, so that you, once you've compressed the information, all subsequent processing steps deal with that level of compression, and it just gets more and more efficient as you go along, and then you will quickly get to binary, you know, on off.
I think I'd have to think a little bit about the answer, but notice that Simon was talking about the second order neurons, which are equivalent of bipolar cells. They're called monopolar cells in flies, and the dynamic range of the photoreceptors themselves is considerably larger. So what these second order neurons are doing is the first synapse essentially taking the signal and amplifying it and matching the gain of that amplification to natural image statistics to make optimal use of subsequent limited neural bandwidth. So you are throwing away some information that's probably available in the photoreceptors, but nonetheless, I think your point about how photoreceptors are going to be optimized in this sort of context is still worth thinking about.
Yeah, looking at frogs that have cone-like rods. When you've flipped, you take what used to be a low dynamic range photoreceptor and massively increase its dynamic range. That should have implications for all this kind of stuff. That would, yeah, I don't know, be interesting. But yeah, there must be selection pressures for this.
On that note, we will head out to the foyer there for some drinks and nibbles, where I hope you all talk to each other as well as to our speakers, but only after they've had a chance to have a drink. But before we do that, please join me in thanking Jolyon and Danny for a very thought-provoking talk.